Uh, Good afternoon, everyone. Welcome to, I guess, the first CIC lecture of the calendar year 2025. Thanks for coming right before the snowstorm. So enjoy your snow day tomorrow, or not, depending on if you have animal work. I don't. Just so you know. Just is that yeah okay. So it gives me great pleasure today to introduce a long-time collaborator, Dr. Clara Moreau. So I've known Clara since she was a PhD student with Dr. Sébastien Jacquemont over at Saint-Justine. And she's done a lot of really cool work since that time on the intersection between, at least in that period, kind of rare CNVs and their implication in an array of psychiatric disorders. She then went on to do a really cool postdoc at USC, which I suspect is what a lot of this will be based on, unless you have a bunch of new work that, so I won't give away the ending. But recently, it's been 4 or 5 months. Has it been 4 or 5 months? I'm doing this all off the top of my head. 5 months. That Clara has gone back to kind of like her scientific home at Saint-Justine as a newly minted PI. So if you get close, she has like a new PI smell. Like it smells like licorice and candy canes. But jokes aside, I'm very happy to have Clara back here to give. This is the second time you've given a lecture, I think. I think you gave one before. Either way, I'm looking forward to this one. And it's nice to have you back here and to see kind of what you did at USC and what you're gonna be doing in your research program now. Welcome.
Thank you very much. Can you hear me? Yes, working. Great. Amazing. So yes, thanks for the introduction, Malar. So I did indeed my PhD with Sébastien Jacquemont, but also Pierre-Lune Bellec. And yes, and after I went to the Pasteur Institute in Paris and then USC with Paul Thompson for the Enigma Consortium. So today, can I use that? So today I'm going to explain how we can combine neuroimaging and genomics to understand biological mechanism underlying psychiatric condition. So, wow, sorry about that. So I will start by addressing, in the introduction, I will start by searching why we are unable today in 2025 to find any reproducible biomarker in psychiatry. Unless you know any psychiatric condition. So why is it the case? So most research have been using a psychiatric first approach, like starting from the diagnosis to understand clinical traits, cognitive dimension, brain endophenotypes, and genetic risk. I'm not going to talk a lot about clinics because that's not really my field, but we all know there is a lot of heterogeneity at the clinical level. It's almost impossible to find a patient with not like additional diagnoses, comorbidities. Like for example, if you take a patient with ASD, this patient will probably have several other diagnoses like ADHD, OCD, anxiety, eating disorder, etc. So it's almost impossible to find a patient with only one diagnosis. And we don't really take them into account in our models. We are doing better today, but most research did not really take that into account. So now, to address that, researchers have been using dimensional approach, subtyping approach. So it's a bit better, but still not working. So why is it the case? So at the brain level, people have been complaining a lot about sample size, method variability. So for sample size, for example, if you look at this Nature paper from 2 years ago, they claim that you need thousands of subjects to perform any reproducible brain-wide association study using structural and fMRI data. So there is something we need to take into account here. It was mainly for traits, not for condition or genetic risk factors. So indeed, if you look at the effect of IQ, you need thousands of subjects. But if you look at and I mean, it really depends what you are looking at, obviously. And now we kind of answer like this issue with all these large data set consortiums that have been built since like in the last decade, let's say. Like Abide was like 2014. Imagine was a bit before, but it was like, it's been 10 years, around 10 years. So, I mean, if you want, you can have a large sample. Obviously, if you do like 20 cases, 20 controls with a subject with autism, even if you match them for sex and age, it's probably not enough. But yeah, we have large data. So now with Enigma, I'm going to talk mainly about fMRI, but Enigma has done a lot with structural data. So we just like last month combined a large data set of 15,000 cases and 20,000 control of subjects with resting state fMRI data from the Enigma consortium for 8 conditions. So ADHD, anorexia, ASD, bipolar, MDD, OCD, PTSD, schizophrenia. So we are processing them right now. It's ongoing, but it's really exciting, and we will be able to address this sample size issue at least. So let's say this issue is, we take care of that. But so we still have a lot of other issues. So like methodological variability. A lot of people complain about that, and it's quite right, especially in fMRI. In structural MRI, it's less a problem, but I mean, it's still a problem, but less an issue. But in fMRI, it's been quite crazy, and a lot of papers have been published about like reproducible crises in fMRI, blah, blah, blah. We don't like fMRI. I mean, I heard that so much. So this Nature paper from 4 years ago from Paul Drack and colleagues, it was quite stressful. So they gave a data set to 70 teams, and they asked them to address, I don't know, a few hypotheses, like 3-4 hypotheses. And they let them pick whatever method they wanted to use. And then they compared results. So that's the correlation maps of the different results. So each row is like a team, each column is a team. So they realized like the 70 teams use 70 different methods to do their analysis. Not stressful at all. And that's the correlation of the result. So it's not, I mean, you can see that a good part of them agree on the result, but some totally disagree. And so it's still not really reassuring. So why is it the case? Is like the group like with anti-correlated with the rest, did they do really bad choices? It's not that easy. There is like many reasons that could explain these differences. So we look like there's so many decisions to take when you do fMRI analysis. So for example, let's say everybody does the same pre-processing, which is already like in the ideal world. It's not the case. Then you have to do like denoising. You have to use different denoising methods. There's tons of different denoising you can do. I will come back on that. But let's say there is only 5 options. Then you have to pick which feature you want to extract. Let's say there is only 5. There is much more than that. And which stats model you want to do. So just that, really simplifying, you already have like 125 way of doing your analysis. So yeah, you have a lot of decision to take to perform your analysis who can lead to different results. So when I was working for Paul Thompson at USC, he asked me to find a way for everybody to run the same things. But I was like, how to pick a right way, because everybody has their way of doing and they have their reason of doing it. There's tons of paper who justify the choice. So I started to ask so many people. So we started to do a survey of like, we ask Enigma experts and users of fMRI. We asked like 60 people across 30 working groups of Enigma about like which pre-processing steps have the greatest potential to change the results, basically meaning which are the most important to be harmonized across groups, to be able to compare something at the end, because we have like those 40,000 subjects with Enigma, which we want sites to process them the same way. So in red, you have like people considering themselves as experts, And in blue, you have like users, fMRI users. So as you can see, expert, I mean, it's a bit small, but here you have like confound removal, motion scrubbing, and GSR. So we ask about every steps, obviously, but here I will just go through a few. So for example, for scrubbing, it was the less complicated one. Scrubbing is like a debate in the field, obviously. But here in this survey, almost everybody agrees that at least it provides benefits, and half of them said it's required. So we said, great, we will all do scrubbing. So now we want to do scrubbing. People say, which type of scrubbing? There's many types of scrubbing. So we are debating on that now. It never ends. But at least we agree we want to do scrubbing. Then GSR. So GSR, everybody know, it's a huge problem in the field. In this survey, it was quite interesting to see that half of them said it should never be done. And the others half said it provides benefit or it's required. And so it really depends which PI you are working with, basically. So there is different school. So basically, we concluded for that, but we kind of knew it. We now have to do both. It's like for structural imaging, you have to do with and without adjusting for global signal like total surface area or mean thickness or whatever. You have to do with and without. So that's the same for GSR. There are like so much discussion about GSR, but we kind of all agree you have to do both. And now for denoising method. So that's another problem. I'm not going to talk about that forever, but we proposed 6 methods. So simple, which is just like motion parameters, derivatives, squared motion parameters, derivatives, ACOM core, IC aroma, ACOM core, IC aroma, simple plus GSR, and IC aroma plus GSR. And we ask like which method is a good denoising method that everybody should use to have like harmonized data. Most experts agree that a simple denoising and ACOM core are good enough to do your analysis. So what we did, what we did is like we ended up building some guidelines that everybody in Enigma is going to run with multiverse analysis, meaning there is not only one pipeline you are going to run, but you are going to run 3 different pipelines to denoise your signal. One with only ACOM core, one with motion parameters, what I call simple, plus scrubbing, and one plus GSR on top of it. Then there is a lot of debates about like which features you can extract, which atlases are you using, the Schaeffer's, whatever. There's tons of different atlases, the Buckner, whatever. So we also asked about that, and we all agree that we will use Schaeffer combined with Free Surfer and the Buckner 2011 for the cerebellum and Free Surfer for subcortical. And we will do connectomoid analysis and functional gradients. We'll do only subcortical region for seed-based connectivity. And people asked to add FLFF and RIO. So we added that. We also had like several quality control standards that we want to implement. So for now, we have the ME/FD at 0.5, but we will have other thresholds. So that's like a lot of methodological discussion. We can talk about it later, but I just wanted to say like about this triangle, let's say this has been kind of addressed for fMRI, let's say. It's not fully addressed, obviously, but we are going toward a more harmonized method. And as you understand there is not a good way of doing it, we will do multiverse analysis. So then let's go to the genetic level. Let's say everything is addressed in clinics at the clinical and neuroimaging level. So at the genetic level, why do we not have clear biomarker? So there is 2 different mechanisms that are important to understand here. It's like polygenicity and pleiotropy. Polygenicity simply means that you have thousands of genes involved all over the genome associated with each conditions. So if we take like NDDs, neurodevelopmental disorders. You have in the study 1,500 high confidence genes associated with NDDs, and on top of it, 6,000 candidate genes. And they are located all over the genome. So there is no, as in MRI, there is no brain region associated with one condition. In genetics, there is no chromosome or part of a chromosome associated with any condition. It's everywhere. And to add complexity, there is pleiotropy. So pleiotropy means that there is one genetic mutation that will confer risk for several conditions. So most of them are not uniquely associated with one condition. I mean, almost all of them. They confer risk for many other conditions. So for example, you have genetic mutations that confer risk for autism, some that confer risk for schizophrenia, and you have like a bunch that are in commons. While if you think about it, you will think like ASD and schizophrenia have nothing in common, but at the genetic level, they do. So that's for rare variant, but for common variants, that has been extensively done by the Psychiatric Genomic Consortium. So for example, in this like large study, they correlated GWAS result across like 8 conditions. And they found like 3 clusters of interrelated disorder at the genomic level, SNP-based level. So mood and psychotic disorder, disorder with compulsive behavior, and early onset neurodevelopmental disorders. So that kind of illustrates the pleiotropy problem that could explain why we don't find a clear biomarker underlying a condition. And so these 2 concepts, I like to summarize them with this like 3 musketeers sentence: one for all for one. One for all is standing for pleiotropy, one mutation conferring risk for several conditions. All for one for polygenicity, several conditions conferring risk for the same, several mutations conferring risk for the same psychiatric condition. Okay, so we are here now. A lot of problem at many levels, and obviously I didn't talk about everything. But so what can we do for the genetics? So one way of addressing that is to start from the genomes to understand, instead of going from the diagnostic, you start from the genetics with a bottom-up approach to try to identify imaging dimension, cognitive dimension, and going back to the clinics, but not to the diagnosis. That's really not the point here. So how can we do that? There's different genetic risk factors we can use, but we need genetic risk factors that are frequent enough to be able to build a cohort. So that's the frequency, but rare enough to have a decent effect size. You don't want you don't want SNP, single nucleotide polymorphism, because their effect is so tiny that you will not be able to do much with that. But you don't want extremely rare condition genetic risk factors that are impossible to find. You have 1 case every year in Canada. So we decided to go with CNVs, which are copy number variation. They are a deletion or duplication of a DNA segment that typically encompass several genes, and they are present in less than 1% of the general population. But at St. Justine, for example, we have like 10 to 15% of the children who carry such mutation. So what's interesting is like if you build cohorts of patients who carry this mutation, you can find a really much bigger effect size on different brain endophenotypes. So here it's on connectivity, but we did that on structure, brain structure, and now like we have students doing that on EEG. And we keep seeing the same, like the effect size of this genetic risk factor are much higher than the one associated with like ADHD or ASD. Schizophrenia has a bit better effect size. So that could suggest an homogeneity of the signal that is due to the biological homogeneity. So with that, we wanted to address one. We had 2 hypotheses, and we wanted to ask about the converging patterns. So we have like 2 scenarios. The first one is this one, where like we said we have 1,000 genetic risk factors for ASD, let's say, but it's an example. They will give us like 1,000 forms of autism. So each each child carrying one mutation is going to be totally different, while the alternative scenario is like we expect to see some convergence from the genome to the phenome. At the end, you will obviously not have one form of ASD, maybe 10 or 20 or more, but not 1,000. So we expect some convergence. So to test this hypothesis, we aggregated like many data sets from different hospitals with like CNVs carriers, and we characterize their impact on functional connectivity. So that's what is reported here on the left side, and in the middle you have like brain networks. And we wanted to see if we are able to find any convergence across these different genetic risk factors. And so that's what we did. And so in red, you have overconnectivity. In blue, you have underconnectivity. And we were looking for, as I said, convergence. So first, what you can see is like every network is impacted by these genetic risk factors. So there is differences everywhere. But we found some convergence as well. So for example, if you zoom on the basal ganglia thalamus network, you can see some similarities across genetic risk factors. So that would be that represents the pattern of the thalamus. So that was like the thalamus was mainly overconnected with the rest of the brain, but mainly with like sensory motor area. So for example, if you look for ASD, the thalamus is overconnected with motor, auditory, visual area, and it was quite similar for schizophrenia, neuroticism in general population, deletion 16p11.2 and duplication 22q11.2. And then something we can also see at the organoid level, if like we had like some colleagues doing 22q organoid deletion organoids, and they also found like abnormal outgrowth and the increased spontaneous activity of the thalamocortical axons. So that could mirror what we observe at the connectivity level. I mean, we could be mirroring what they observe at the cellular level, maybe. But so more globally, I would like to put this result in a larger theoretical framework. So the idea here, so before you saw like the little triangles, but we expanded it. And so now we want to still use genetics to identify brain dimension that could reflect shared behaviors that are observed across different conditions. So the hypothesis is like this genetic risk factor, in our case that I just explained, they are going to converge on similar molecular pathway. So I will give example later. That could, for example, converge on some like brain signatures. So this thalamosensory motor overconnectivity being one of them. And this like thalamosensory motor connectivity could be reflected by sensory motor impairment at the clinical level, which is something that is not specific to one condition. That's a cognitive dimension that is observed across different disorders. So that would go beyond like doing cases versus controls at the clinical level. So how can we test that, and how can we go further with this like theoretical framework? So there is a different way at every level. So for example, at the genetic level, we can, as I said, like we can cluster the genetic mutation based on their function. So that's something I did at the Pasteur Institute. So for example, we know that many mutations involved in autism are involved at the synaptic level. So for example, if we take the Shank family, Shank is like a highly associated mutation with autism. So, and if you look at its expression profile, it's mainly expressed in the cerebellum. And so we looked at many patients with shrink-free mutation at Robert Debré Hospital in Paris. And I mean, I took the most extreme case, but there is definitely an effect on the cerebellum. I mean, in that case, it's quite extreme, but it's just to illustrate what we are looking at. Another way is to annotate genes based on burden scores. So how does it work? That's something I was doing with Sébastien Jacquemont at Saint-Justine. So instead of doing like one CNV at a time, you can annotate genes within each CNVs to use like continuous score. So like that, you can include any carriers of any CNVs in your analysis. So you go with like those burden scores and not doing one CNV at a time. So we performed preliminary analysis on that using the PLI score, which is the probability of being loss of function intolerant for genes. And it was the best predictor of IQ loss. And so we found again that one of the most affected region was the thalamus. And then the connectivity profile was quite complex. But yeah, that was like some preliminary result we had for going, I mean, it's more to illustrate the general principle of going beyond the case versus control at the genetic level. So then there is like, we can combine this bottom-up approach with a top-down approach doing transdiagnosis analysis. So that's something we are doing now with Enigma, as you can expect with all these data set I presented in the introduction. But so for something we did before is like to correlate brain signature at the connectivity level of, I don't remember how many conditions, but maybe like 6 conditions at the connectivity level. So that represents the distribution of correlation across pairs of conditions. So for example, you can see a good correlation between schizophrenia and bipolar, schizophrenia and ASD, etc. So we wanted to see if this correlation obtained at the connectivity level could mirror what has been done at the genetic level by the PGC. So that was this like figure I presented in the introduction where the correlated GWAS summaries. So in the x-axis, you have the connectivity correlation. And in the y-axis, you have the genetic correlation. So you can see a good concordance between our observation at the brain level and their observation at the genetic level in terms of overlap. So for example, our like 52, our R equals 52 correlation between schizophrenia and bipolar was quite similar with the observation at the genetic level. And our like 0.3 correlation between ASD and schizophrenia was also quite similar with their observation at the genetic level. So we obtained a concordance of 70%, which was quite impressive because it really means that connectivity, I mean, brain endophenotypes are following genetics observation. We also compared with transcriptomic results with this science paper by Gundal and colleague, and similarly, we found a good degree of concordance between our observation, so the same as before, and their observation at the gene expression level. So it seems like there is the stability of the overlap across condition and across different level of observation. So at the genomic, transcriptomic, and connectomic level. It also means that functional connectivity is not only noise, it seems to be a good phenotypes to study psychiatry. So we also looked at brain anatomy and specifically for one condition. So we looked at thickness and surface area. And during my first postdoc, I mainly worked on anorexia and this relationship with other conditions. And when I started working on anorexia nervosa, I was really impressed by the lack of interest in this condition. So it's largely understudied if you look at anorexia versus other conditions. So that's obviously is Alzheimer, but that's schizophrenia, autism, etc. So that's the number of publications by year. While anorexia has the highest lifetime mortality rate of all psychiatric condition, and relapse is the norm. You have like 1/3 of the patient will go back to the hospital after being treated. So I was really impressed. There is not much research about that. So I had access to a cohort of 124 cases and 112 patients of patient with early onset form of anorexia at Robert Debré Hospital in Paris. So we started simply by characterizing the effect on cortical thickness, subcortical volume, surface area. We found a really large effect on cortical thickness that we compared with other conditions from the Enigma consortium. And as you can see that the effect sizes distribution, there is like a lot of, like the anorexia has a larger effect size, higher effect size. So we found like this large reduction of cortical thickness in anorexia with CSF increase and subcortical volume decrease as well. And then we compared that with this other condition. So we correlated maps, and we found a really good correlation with OCD, which kind of mirrors the clinics because we know that's one of the main comorbidity of anorexia. Also schizophrenia, which is more surprising, and bipolar, and it wasn't undercorrelated with ADHD. Then again, we compared that with genetic findings. So we took like the Nature Genetics GWAS that has been done a few years ago, and where they compared results in anorexia with other conditions. So we did the same, and we compared our result with their results. And again, we found a good degree of concordance, not as good as before, but they also found a good correlation with OCD that was here, which is here now. That's genetic correlation and brain-based correlation. MDD and schizophrenia. So at the genetic level, they tend to also find a significant correlation with schizophrenia, which is something we observed at the brain level. So we are now pursuing this analysis on a much larger data set. So we obtained a R1 last year on anorexia with Paul Thompson and Stefan Ehrlich. So we aggregated this amount of data. So I'm now leading this fMRI analysis for the Enigma consortium. So we have like 800 cases and 1,000 controls for anorexia, and we want to do multimodal analysis for anorexia. And obviously then we want to continue that by comparing with other conditions as I did before, but on a much smaller data set and on structural data. So with the data I showed you before, we are going to compare anorexia, which is here. That's what I just showed you, with these 7 other psychiatric conditions. So that's something that is ongoing. So it's going to take a few years, I guess. And more at Saint-Justine now, I am like starting to acquire data of like patients with anorexia and other ARFID condition. I'm going to talk about that just now. And one of the hypotheses we would like to test is like, are we able to predict the probability of relapse of patients with anorexia? Earlier, I told you we have 1/3 of the patients who relapse after body weight restoration. And we have no predictor of that. And something we can observe at the brain level on the Paris data is like the brain signal evolved after body weight restoration. So that's the patient that are at like -3 standard deviation for their BMI, and that's after almost total weight restoration. So some patients have no abnormality after weight restoration, and some patients still have some abnormalities. So we would like to have this degree of brain restoration to try to predict the probability of relapse. As we know, there is like 30% of them that are coming back, that are going to come back. So that's one project we are starting at Saint-Justine, and we will also do that with, like, in mirror with the Paris Hospital Robert Debré. So yeah, we want to ask his brain restoration level can predict the probability of relapse. We have another project with ARFID. So one of the questions we had, again, with that we can address with transdiagnostic approach. So I always wonder, like in anorexia, you have like 9 women for one man. And I was wondering if there is like a bias in the diagnosis. So for example, if a young patient, a male patient, present eating traits and social interaction impairment, this patient is most likely to be diagnosed with autism. And we know that like patients with autism do have picky eating behavior and sensory aversions. And we also know that young girls with anorexia also present sensory interaction impairment, but they will primarily be diagnosed with anorexia. That could also be due to like the age, like sex and age, because like there's like these like developmental differences, especially around like 10, 12 years old between girls and boys. So we know that. So now there is this new diagnosis that has been introduced in the DSM-5 and the ICD-11, which is ARFID, meaning avoidant restrictive food intake disorder. It's basically a diagnosis where they put everything they don't really understand. So it's like when a patient do not eat, but it's not because it's not motivated by body image concern. I mean, if we follow the narrative of the patient, of fear of gaining weight, it's motivated by a lack of interest in food or fear of aversive somatic consequences of food intake, like vomiting, or in sensory sensitivity impairment, which could, for me, be autism, but not only for me. So there's a mix of things in ARFID, in this new ARFID diagnosis. And obviously you have more boys in this diagnosis, and they are also a bit younger. And so we started to collect data with ARFID. We have like almost 50 cases now. And we found a really different brain signature of ARFID compared to anorexia, which is much closer to ASD, obviously. So that's something we want to continue. And I would like to also compare them with the CNV carriers, because the 16p11.2, for example, the deletion is associated with autism, as I mentioned earlier, but it's also associated with obesity, while the duplication is associated with schizophrenia and anorexia. So there are kind of good genetic models of eating disorders. It's similar for 22q11.2. So we want to understand what is due to BMI, what is due to sensory impairment, what is due to sex, to age. Like, it's kind of messy, and doing this transdiagnostic analysis could help to address that. And finally, but that I haven't started that, but that's more like what I would like to do in the future, like to integrate more like multi-organ data to go beyond the brain, because similarly, when I worked in different hospitals, I was always impressed, like when you like follow patients, you take care only about the brain and their cognition, but they often have tons of other stuff that we don't really care about because we are not working on that. But it's quite important for them. So, for example, patients with ASD, they, as I said, they have a higher probability of having like tons of psychiatric comorbidities, but they also have like other things like gastrointestinal issues, cardiometabolic disease, and sleep issues, immune disease, etc. So that's something we are not taking into account. And similarly, the CNV risk factor for psychiatric condition, indeed, they confer risk for autism and whatever psychiatric condition, but they also confer risk for different medical conditions. So, for example, if you look at the 16p11.2 CNV in PubMed, you will find, okay, risk for autism, blah, 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 but also a lot of paper about cardiometabolic disease. So when I was doing my PhD, I was like, what is that about? It's not really what I'm working on. And I don't want to read that, but I mean, it's what they have, so we should read that. I mean, I should. So I would like to be able to collect more of these data to like understand the patient like more like entirely, and not only like their brain and cognitive scores. And I don't know if you follow this paper published in Nature Medicine by the Zaleski group. So it was quite interesting where they compared in psychiatric condition the organ theoretical age versus actual biological age. It was done in UK Biobank, and they followed like 8 systems, including the brain, obviously. And they compared the biological aging of different organs in psychiatric conditions. So that's the 8 different, I mean, I don't know, there's more than 8, the different systems it compares. And the brain was really not, where is the brain? The brain is here and here, was really not like the one with more like with this accelerated biological aging compared to different organs. And they did that for different conditions. So obviously we don't know what the cause, what the consequences, because like maybe this all this like accelerating aging is because of the condition where you put like yourself in a specific environment, you eat specific type of food, blah, blah, blah. And at the end, you have all the body that is impacted. But that's not the primary cause. So we don't know that, obviously. But it's still interesting to see how we can use this data. So for example, for this project, to not use only the brain restoration level, but different data from the whole body to predict this probability of relapse. We also want to collect, obviously, genetic data in this patient because there are some genetic risk factors for anorexia that we know. There are still only like risk factors, but still interesting to get to because we expect that the one carrying genetic risk factor will have a higher risk of relapse. So done, and that's just like summary. That's the theoretical framework I've been presenting, and it's different way of addressing this lack of biomarker. I guess we'll never find biomarker for any of these conditions, but we can be able to find other interesting things. I can go back on this slide. So as Mara said, just open my labs. So it's my new students. And I would like to thank participants, funding agencies, and Enigma working groups, my colleagues from everywhere, and questions.
What's either me or
try.
Yashar? Yeah, it was either one of us. Thank you for giving this talk. It was just great to see multi-scale, multi-dimension approaches being combined in this way. My question is on going back to some of the results you presented on the increased connectivity of the thalamus. And in RFID, you mentioned that you're interested in examining sensory and somatic manifestation of possible biological mechanisms. And I would like to give you the chance or ask you to expand on your future directions, what you would like to look at, because the thalamus is a big sensory hub, and I feel like these two combined together might open the door for a lot of different examination in neurodevelopmental disorders. For example, in OCD, we see a lot of sensory issues. In ASD, we see a lot of sensory issues. So it's starting with RFID, but expanding on it. What do you expect to see increased in specific disorders or decreases in other disorders?
Yeah, so for RFID, we just started, so we have 50 cases only on structural data, but and we are the first imaging study, so published, ongoing. But we want to do fMRI analysis and we expect to find this similar thalamo sensory motor overconnectivity pattern. So that's what I would expect because, yeah, they also have comorbidities with OCD, ADHD, ASD, so we expect that. And so I don't have a clear expectation about I don't know which part of the thalamus or stuff like that. That's something we'll probably do in a second step, with better, a specific approach on the thalamus. But thalamus here was one of the example in this theoretical framework. That's basically to illustrate, we are looking for these general dimensions that are spanning across the different diagnoses. So we are not looking for markers of one diagnostic, where we are looking for markers of dimensions that are driven by the biology. So I don't know if it's clear. It's quite complicated to explain every time. But yeah, and this thalamosensory motor overconnectivity, the hypothesis here is like it could reflect sensory motor impairment. But even here, we were not able to test it so far because, I mean, for the data we are collecting, it's easy. But for the rest of the data, most of the time they did not collect the same scale, the same same test. Like each diagnosis have their own battery tests, and it's quite difficult to harmonize. But that's why we like RDoc now. And in UK Biobank, you have this type of scores and ABCD, but all these psychiatry first cohort, they only have I don't know, for ASD, they will have ADOS, RCRS, blah, 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 for ADHD, other things. But in ADHD, you will not have SRS and these type of things. So it's quite difficult to verify this hypothesis at the behavioral level. But that's where we are, that's what we would like to do, yeah. Are you working on ARFID? Do you know anything about ARFID?
I'm not working on ARFID,
No,
but
because nobody know about it.
yeah, we have a big cohort here at the Douglas. And funny enough, 2 speakers ago was on anorexia nervosa, and we touched on a lot of the cohorts that are being collected here. I think some of them have multimodal imaging. I'm just not sure if it matches what you're working with.
But I know Linda Boyle is collecting
exactly
data. Yeah.
That's exactly what I'm talking about. Yeah.
Yeah, I know. Okay, great. But yeah, she's not working on ARFID. I mean, that's what she said. But
yeah, not
it would be interesting.
ARFID, but I think they have, like you said, what you don't understand, you put it in ARFID. There are a lot of these diagnoses, and then there's ARFID.
Yeah.
Kind of like the other category.
Oh, it's a comorbidity. Like in ASD,
yeah,
I say many cases they have eating disorders, and we just put just ASD.
Yeah, exactly. Yeah, thanks a lot.
Is anyone else know about ARFID? Yes. Everybody?
Not me. Thanks for the talk and really impressive work. I should say I really appreciate this, putting aside the diagnosis and going, and I really agree, it's really nice work. But as you said at the end, maybe we won't get biomarkers, because when you don't have labels, what do you work with? And that's what I want to know your thoughts, because we are looking at mechanisms, convergence, divergence, and you can only look at that when you put those correlations with genetic correlation between these labels that we don't believe in, but then we use them to look. If you want to make it a continuous, and that's what I've been thinking for a while, if we want to make that continuous, how do we go for a continuous measure of our connectivity changes across subjects? And what is on the other axis that we look at the correlation? What do we correlate these biological brain measures to if we don't use the labels?
I mean, ideally is what we were discussing, discussion. Ideally, we would use these cognitive scores that would go beyond the diagnosis, but we don't have them harmonized across condition. So I would say now, new cohorts, they do collect more homogeneous scores that we will be able to compare and put on the y-axis instead of. But for example, in the plot, where is it? Yeah. So here, I did not talk about it, but we also tested neuroticism and fluid intelligence in the general population. So that's something that is not a diagnosis. And we found negative correlation between autism and fluid intelligence, ADHD, fluid intelligence, etc. So that's not ideal, but those scores maybe beyond these two metrics can help addressing this problem. I totally agree. It's a bit weird to say we don't believe in this diagnosis, but then we do that. But I still think it's interesting because it shows still how overlapping they are and how we have to rebuild, like we have to work on this specific cluster, like anorexia, ARFID, ASD. We know like there's a lot of relationship across them. So it will help us to reconceptualize this diagnosis.
I think one of the problem is the questionnaire, the cognitive measures
Yeah.
are designed to diagnose
Yes,
people.
exactly.
So the range is like a bimodal rather than a normal distribution
Yeah.
which you want there. And then another question is, you said resting state or fMRI connectivity is not noise, and I agree.
Is
I
not
don't
what?
believe it's not noise.
Yes.
I agree, it's not noise.
Thank you.
But how do we
But-
bring it back? We are putting correlations between 100, 1000 by 1000 matrices across population, and we get one value across all of that. And then you try, I have tried with that, with a structural, like whatever you put there. You try to bring it back. There's so, like all of a sudden you have 1 point estimate that corresponds to so much variability on the other side. When you want to bring it to subject, each of them is having a matrix itself. And I think that's why we don't have biomarker for anything, because it's just, if it's a continuous thing, it really depends where you fall. If you're really extreme, maybe it works for that particular subject, but we cannot parse that multidimensional space. What can we do with these connectivity matrices at the end when we use them? Like, how do we bring that to a biology that we can act upon for patients?
Like you act up on, like you want to
Like I'm
do
saying, like
intervention?
you were showing that we go, instead of, now we go from copy number variance to genomics to the,
Yeah.
and we have the brain in the middle.
Yeah.
And my understanding, you might say, no, that's not even what we want to do. My understanding is like, if we want to go towards drug discovery or help or intervention,
Mhm.
that's our point of entrance,
Yeah,
the
yeah,
brain.
yeah.
We are not changing people's genetics. Maybe we will, I don't know. but let's say we want to work on brain. Now we have this like condensed measure of 1000 by
Yeah, yeah, yeah.
1000 like regions.
Okay, I understand. Yeah. Well, for now, I would not say that's a goal. I would say that's a really long-term goal. And I would say first we want to position our patient on those like continuum and use genetics and brain data to help us on that. And then I think when we will be at that stage, meaning in 10 or 20 years, maybe, we will maybe come back to understand what's the brain features or genetic features we could have an impact on. But I think that will be like later, like we will reverse and come back. So right now we are going up. When I said going like from the diagnosis, it's like to do transdiagnostic analysis and to better understand it, but we are going up. That's the bottom-up approach. And I think when we will be done identifying this dimension, like we say sensory motor impairment, but other things, and even, anyway, we might go back to like the molecular pathway, let's say, to try to see if we can target anything, or even like with psychotherapy intervention like that, we might be able to do some intervention that could maybe address not a diagnosis, but this specific dimension.
Thank you. Like, I'll
There's
talk to you
tons of
later for
more.
more questions, but it's really interesting work. Thanks.
Thank you very much.
Hey, very interesting work. If you go back to the graph that we were just on with the two correlations, and like the X and Y X. Yeah.
Sorry.
Yeah, exactly. So you did this with connectivity here, and I'm wondering if you did that also with morphometry on the X-axis, so with like or like atrophy, for example, with the instead of connectivity.
Oh, did I do that?
Yeah.
Yes. Well, not me, but some colleagues did that. And I mean, we did that for anorexia, which is here. That's on structural MRI, thickness, surface, and and subcortical volume. But here, like this graph has been done by, like for example, the Enigma consortium. They compared like these different, like they compared at the sickness level and subcortical volumes, pairs of condition and genetic results, so the same genetic results. And yeah, the concordance was quite similar.
Okay, there's also a concordance in
Yes.
morphometry. Okay, interesting.
Diffusion, no, I think it has not been done.
Okay, interesting.
And EEG, not either. I mean, I can hear you, but
Thank you. Great talk. I mean, so incredible to see kind of all these levels of biology kind of come together in one kind of unifying framework. I think one thing that always interests me and kind of worries me, if I think of something like, I see the maps that you show for the Enigma consortium, like the
I
international
was
maps.
surprised to not have question about that. Yeah.
So, I mean, one, obviously there's a lot of not lit up areas, which is somewhat predictable, right? But the second thing is that, for a lot of these diagnoses, if I take ADHD, for example, right? Like the way ADHD is talked about clinically here, it's not at all what we talk about in Europe, right?
Yeah, that's true.
Especially in France or,
Yeah.
there's, and the case management is completely different.
Yeah.
What we consider to be ADHD is completely different. So how do you reconcile all that?
Yeah, no, that's a great question. And the reviewer from the grant asked us stuff like that, obviously.
It wasn't me.
Yeah. Good, I think. No, but it's critical for any diagnosis, and we have many variables like that we are struggling with, obviously. Yeah, so the diagnosis, what help when we have that is like when we can at least look at the tests they use for the diagnosis and match patient based on scores instead of binary diagnoses. So we tend to have this and often, but not always. And we also, when we don't, there is like some way of like transferring some tests to other tests. But like that's like the neuropsychologists know about that, and I don't. But we for each diagnosis there is some test we can harmonize, and so like that maybe some patient do not fall anymore in the diagnosis and some stay. So, and you can do sensitivity analysis excluding those patients who are not anymore there. Okay. So that could help for this specific problem, but there's tons of other.
And so if you go about that correlation that one keeps coming up to, have you looked at if there's overrepresentation from different geographic areas for diagnoses that are correlated?
The what? The
kind of plot we were all talking about before.
Oh, okay, yeah. Here?
Yeah, are most of the schizophrenia and bipolar labels, are they all from Europe, for example? So
that has not been done with Enigma data,
Okay.
but we want to. That has been done like for schizophrenia, it's Cobre, it's
Okay.
Pierre Orban data, it's like aggregation from Montreal plus some public data set. And so it was Abide, ADHD 200,
Yeah.
like all these public data sets, and plus Saint Justine. But now we will obviously do that. And yeah, I mean, with Enigma, when we will have the results. And we should definitely look at that.
Interesting.
Yes.
Even if that's
Very.
true, that's
Yeah, it's
interesting.
interesting. Yeah, definitely. So many. No, but it's really interesting. But like all this, like that's really just. I mean, I just listed like six issues, but there is so many. Like the environmental effect, the age effect, the sex effect. What? Yes. Thomas Paustel asked me a lot about that.
I'm sure.
Yes.
I just want to thank you very much for coming to give the talk and join me in congratulating Clara on just an incredible body of work. Thanks.